we'll talk about the topic of bacterial genetics and variation. So this is a big topic. We won't be able to finish it today, and we will discuss part of it next time on Thursday. Okay, bacterial genetics and variation. When we talk about genetics, we are talking about genetic material. So we'll talk about the DNA, chromosome, genome, genes, so all related terms. And variation, we'll cover it on Thursday. So variation means change. Like in microorganisms, we are talking about mutation. So how you can change the genetic material of a bacteria. So these are the two major parts we'll talk about. Okay, so first we'll talk about what are genes and what are genomes. So every organism has their own genome. So the genome is all the genetic instructions for development of cellular structure, metabolic functions, and their regulation. Here, we need to remember this point. First, genome include all genetic instructions. All means that any kind of genetic instructions. For example, in bacteria, this is a bacterial cell, it includes its chromosome. It's a circular DNA, and it also includes some plasmid in the cytosol. Plasmid are small DNA molecules. And for eukaryotic cells, it's even more complicated. It includes its chromosomes. Like in human cells, we have 46 chromosomes in every cell, and not including sperms and eggs. Every normal cell. And also, there are plasmids. These are the plasmids. And also we have mitochondria. And mitochondria also contains DNA. That's also genetic instructions. And for plant cells, there is a chloroplast. It also contains genetic instructions. And for virus, their genome might be DNA or RNA. All these materials formed the genomes of different organisms. They may play different functions, such as development of cellular structures, metabolism, and regulation of this of the genome. So this is the term of genome we need to remember. The next, we'll talk about genes. So what are genes? We know this term, and almost everyone is familiar with genes, but it's not easy to define a gene, and a different definition might be different. Here we use this definition. So genes are located within chromosomes or plasmids. So it's on either the DNA or RNA, for example, DNA of bacteria, or RNA for some viruses. They are specific fragment located on DNA or RNA. Okay, so that's a pretty simple definition, but the function of different genes are different. For example, for this gene, it's gene 1. it encodes a structural protein. So information from this DNA can be transcribed into mRNA, and then mRNA can be translated into polypeptide. So basically the protein sequence. So this is the first type of gene which encode structural proteins. Or the second gene, it encodes regulatory RNA. So the production of this gene is just RNA, not protein. And for this gene, it encodes a transfer RNA. Okay, this transfer RNA is useful for protein synthesis, but it's only an RNA molecule. It does not translate into protein sequence. So these are genes. They are specific fragments on the chromosomes or plasmids. There is also genes on the plasmids. And as we learn molecular biology and general biology, we learn the information flow in organelles, in regular organelles. We know that the information flows through DNA to RNA, then to protein. From DNA to RNA, this step we call transcription. From RNA to protein, we call translation. So you learn this either from high school or from your molecular biology class. And DNA, it can make new DNA molecules based on its own sequence. And this procedure we call DNA replication. So this procedure, that's the central knowledge or central information of molecular biology, or we call the central dogma of molecular biology. I'd like to show you this video on central dogma. It can help you better understand how the information flow in organelles. Okay. Central dogma.
Double helix contains two linear sequences of the letters A, C, G.
Sorry. I'll increase the volume so that you can hear it.
The DNA double helix contains two linear sequences.
Can you hear the audio? It's paused now. Okay, great. Let's continue.
Sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay, so this video shows us the flow of information from DNA to RNA to protein, or we call the central dogma. But I want to make it clear that this video shows us the central dogma of eukaryotic organisms. Because in the video you can see there is intron in genes. So after transcription, the intron needs to be spliced out to form mature mRNA so that mRNA can be translated. So that's not happening in bacteria, because in bacteria all the genes are continuous. So there is no intron in bacteria. So just to give you a better impression on central dogma, but keeping that in mind, in bacteria there is no intron. Okay, so let's see. Continue our lecture. The first thing we are learning here is the bacterial genome, and we'll learn the components of bacterial genome. First of all, it contains a bacterial chromosome. Chromosome is a long single molecule of double-stranded circular DNA. So here are some properties of this circular DNA. First, it's a circular DNA, not linear. It's long. Long means relatively long. It's longer than the plasmid, but it's a single molecule. Even though it's very big, it's a single molecule, and it's double-stranded. Here are some properties of a bacterial genome. First, it's smaller than eukaryotic chromosomes. Smaller than eukaryotic chromosomes. So that's when we say big or small, it's a relative definition. Smaller than eukaryotic chromosomes, and it has operons in the chromosome. The operon is a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. I think you learned this on your molecular biology class. So that's a specific structure in bacteria. So there might be several genes linked together, maybe A, B, C, D, 4 genes or 5 genes linked together end to head. Then on upstream of those genes, there is one single promoter, and the single promoter can turn on expression of all these genes. So those genes will be expressed together. So this is an operon. And in the genes, in the structural genes, there is no intron. That's what we mentioned. In the bacterial genes, there is no intron. So there is no RNA splicing procedure in bacteria, in prokaryotic cells. Here is the example of the bacterial genome, bacterial chromosome. This is the E. coli chromosome. I think you can see it clearly here. The number here is 4 million. So it indicates that the E. coli chromosome is over 4 million. This is 0. 0 to here, 4 million, and even longer. It's over 4 million. So this is the size of E. coli chromosome. Okay? And it carries thousands of genes on this this single molecule. And you know, we know that E. coli cells are pretty small under microscope, but you can imagine that this single chromosome is packed tightly in a small single cell. And we showed this picture before. When the bacteria is dead, its chromosome is released outside, and you can see that the chromosome is much longer than the bacterial cell itself. So this is the bacterial E. coli chromosome. Okay, next we'll talk about another component of bacterial genome. That's the plasmid. Plasmid are small DNA molecules. Here small is relative to chromosome. They are extra chromosomal genetic elements capable of autonomous replication. So it's self-replication. Here extra chromosomal means that they are not on chromosomes. So here is the bacterial chromosome, bacterial DNA, and the plasmids are outside. It's in this bacterial cell, but not on the chromosome. And they are also genetic elements. They are also DNA molecules. And they can replicate by themselves, capable of autonomous replication. So they can split and duplicate themselves in the cell to make a lot of copies. So these are the plasmids. Properties of plasmid. First, they can replicate autonomously. So autonomous replication. Second, plasmids are not essential for survival of bacteria. This is what we mentioned previously. The plasmids can carry some genes which can contribute some property to the bacteria, but they are not essential for the survival of bacteria. If the bacteria somehow lose some plasmid, they can still survive. They won't die. The plasmid carry genetic information that provides some properties for the bacteria, because it carries some gene on the plasmid, and those genes can contribute some properties for the bacteria. For example, antibiotic resistance, virulence, or some other properties. And transfer property means that this plasmid can go from one bacteria to another. So it can transfer from one bacteria to another. This is a transfer property. They can be conjugative or non-conjugative. This is plasmid. Let's have some example of plasmid. This is just one schematic structure of a plasmid. It's also a circular DNA. And on this plasmid, it has antibiotic resistance gene, this pink one. and there is some promoter to drive expression of some target gene. And here, this one is a target gene. And here is a real map of one plasmid called PBR322. And this plasmid, I want you to pay attention to the size. You can see the size is 4,361 base pair. So compared to the size of a chromosome, is much smaller. So by E. coli chromosome is over 4 million, and this is only 4,000 base pair. So it's much smaller than a chromosome. Different types of plasmids. The plasmids can be transferred from one cell to another. If they can be transferred, we call this plasmid a conjugative plasmid, or we call it non-conjugative plasmid. And if they can contribute to some phenotypic effects, I mean the plasmid can provide some phenotypic change, and based on this, the plasmid can be a fertility plasmid, or F factor, what we learned before, or bacterial synergenic plasmid, or resistance plasmid. For the fertility plasmid, or we call it F factor, fertility F factor, it confers on cells the ability to transfer bacterial chromosome genes to recipient cells. So it can carry part of the chromosome genes to another cell so that it can transfer the chromosome genes. For resistance plasmid, it carry antibiotic resistance genes, so that the bacteria carrying the plasmid will be antibiotic resistant. Bacterial synergenic plasmid code for substances that kill other bacteria, and these substances we call bacteriocins. Bacteriocins. It can kill other bacteria but does not kill itself. One example of the bacteriocins are the colicin, which is produced by E. coli, which can kill other bacteria but safe for E. coli. So these are different types of bacteria. Another component we'll talk about here is the transposable element. Transposable, according to the name, you can see that they can move. Transposable element. These are segments of DNA. These are segments of DNA that are able to move from one location to another. They are not independent DNA molecule. They are residing on the chromosome or on the plasmid, just one segment of the DNA. And this movement is random. Even though there is a recognition sequence or specific sequence they can recognize, but this movement is random. You can jump on the chromosome from one place to another. And transposable elements are not capable for self-replication. They are not a replicon, so they cannot replicate all by themselves. They are just one piece of DNA on the chromosome or on the plasmid. And transposition is mediated by a site-specific recombination enzyme. And this enzyme we call transposase. It's an enzyme with specific function. Site-specific recombination enzyme, transposase. transposition may be accompanied by duplication. This means that when the transposable element move from one place to another, sometimes they can make a copy and just make a copy of themselves and paste it to another place. So you can say this procedure copy and paste procedure. Here is we'll talk about different types of transposable element. The first type we are talking about is just the simple insertion sequence. This is the most simple structure of a transposable element. It only contains two inverted repeats on the two ends, and in the middle, it only carries one gene, the transposase gene. Very simple structure. We call the insertion sequence, short for IS. Insertion sequences, IS. These are the elements that carry no other genes except the one for transposition. It only carries the transposase gene, but no other genes. The structure is pretty simple, flanking inverted repeats. Importance of transposable this insertion sequence is that we can use it to make insertional mutation. So you can put this element to the bacteria so that they can randomly insert themselves into genes, into the chromosome, so that the genes disrupted will be inactivated. So you make a mutation. And with one single experiment, you can make a lot of mutations. You can make a library of mutations. So this is the first type of transposable element. And of course, there are other types of transposable element. For example, transposons. Transposons are more complicated. The structure is more complicated than this insertion sequence. These are the elements that carry other genes in addition to those involved in transposition. And we learned that this IS structure is flanking repeats with transposase. But for transposon, it also carries other genes. For example, resistance gene. The structure, it's composite transposons. Importance of this transposon is that they can make the bacteria antibiotic resistant. So this is the transposon structure. Here, this picture shows us how this transposon insert into the host DNA. This is a transposon structure. In the middle is the genes they carry. On the two ends, they carry the inverted repeats. And on the host DNA, there is a target site shown in the red color. It's some specific sequence, ATGCA, so that the transposon can recognize this sequence. After recognition, the transposon will cut this red part open, cut from here and here, so that it can make a nick on the DNA, open the DNA, and the transposon is inserted in the middle. But at this time, there are gaps on the two sides, here and here. There are gaps. And the bacterial enzymes will fill in the gaps to fuse the DNA, to make the DNA link together. And this, after this procedure, this transposon will be inserted into the DNA. And also, the red part has two copies now. Here's one copy, here's another copy. So these are how the transposons work. Before we take a break, I'd like to show you guys a short video on transposons, and we'll learn how the DNA transposable elements move on the host DNA.
Transposable elements, which are found in virtually all cells, are segments of DNA that can move from one location in the genome to a target sequence in another. A simple type of transposable element called an insertion sequence or IS element is only 700 to 1500 base pairs long and consists of two basic parts, a coding region for an enzyme called transposase and two short inverted repeat flanking sequences. The cellular machinery transcribes the transposase coding region and translates it into the transposase enzyme. Transposase catalyzes the transposition process. The transposase randomly selects one of many possible target sequences where it will move the insertion element. The IS element, depending on the particular type, may enter the target sequence by one of two general mechanisms: replicative transposition or non-replicative transposition. In replicative transposition, transposase nicks the transposable element and the nicked ends attack the target sequence, making a staggered cut in the target DNA. DNA polymerase fills in the large gaps, and DNA ligase seals the remaining nicks. A recombination event resolves the two molecules. As a result of replicative transposition, the cell has a copy of the transposable element flanked by duplicated target sequences. Non-replicative transposition is sometimes called cut-and-paste replication. In this process, transposase brings inverted repeats and flanking DNA together. One phosphodiester bond is cleaved on each strand at opposite ends of the IS element, yielding free 3' OH groups. Each 3' OH end attacks the other strand of the double helix, creating a new bond that results in hairpin structures. Host carrier DNA is ejected, and the hairpins on the IS element are re-nicked and attack target DNA. From the 3' hydroxyl groups, the cell's DNA polymerase molecules fill in the gaps, and DNA ligase seals the remaining nicks. In this way, the element jumps from one DNA molecule to another. To summarize, in non-replicative transposition, the transposable element is cut and pasted from one DNA molecule to another. In replicative transposition, the transposable element is copied to yield two transposable elements, one in the original position and one in the target DNA.
Another subject we want to talk about is bacteriophage. I think most of you heard of the term before. Bacteriophages are the viruses that infect bacterial cells. So this is the major point. So keep in mind what they really are. So they are viruses. And now our first lecture we introduce viruses a little bit. And we know that they are not cellular organisms. They are just particles containing either DNA or RNA as their genetic material, and also a protein coat as a capsid. So they are obligate intracellular parasites that multiply inside the bacteria by using the bacterial biosynthetic machinery. So a phage or bacteriophage cannot replicate by itself. It can only replicate in the bacteria. So that's because they are virus particles. Okay. Here I will show you the structure of one bacteriophage. Actually, we are more familiar with this T4 phage. Looks like this. It contains one head, a head structure, which is a cubic structure. Its DNA is inside the capsid, and also contains the tail structure. And also there are tail fibers and base plate. So this structure is pretty fancy. So that is why we are more familiar with this complicated structure of phages. But except for this T4 phage, there are also other types of phage showing different structure. Composition of these phages include first nucleic acid. It's either DNA or RNA as their genetic material. And also proteins. These proteins can provide protection for their genome. And also the proteins are useful during infection because they need the proteins to recognize the host cell. So this is the composition and also structure of phage. Here, as I mentioned that we are more familiar with this T4 phage. This T4 phage because they are pretty. But there are also other types of phages like T3 phage. It's also a double-strand DNA phage, but there is only one head. The tail is very short, only 20 nanometer. Very short tail. While for T4 phage, the tail is 110 nanometer. It's pretty long. While for T1 phage, the tail is even longer, but there is no fibers and no tail plate. So a different structure. And also there are these helical phages. This is the one we learned. This is one plant phage, which this is one helical phage which can infect other bacteria. So this is M13 phage, which can infect other bacterial cells. Okay, let's watch this video about T4 phage. In this video, you will see this fancy structure can fly and then fall down onto the host cell. It's much similar like a satellite or a spacecraft.
The T4 phage initiates infection of an E. coli bacterium by recognizing cell surface receptors of the host with its long tail fibers. A recognition signal is sent through the long tail fibers to the base plate. This unravels the short tail fibers that bind irreversibly to the E. coli cell surface. The base plate changes conformation and the tail sheath contracts, causing GP5 at the end of the tail tube to puncture the outer membrane of the cell. The lysosome domain of GP5 is activated and degrades the periplasmic peptidoglycan layer. The remaining part of the membrane is degraded and DNA from the head of the phage can travel through the tail tube and enter the E. coli. After the life cycle is complete, the host cell bursts open and ejects the newly built viruses into the environment, destroying the host cell.
Okay, so basically this is a procedure how a T4 phage infect the E. coli cell and replicate inside. And later we'll talk about different types of phages, and some of them will kill the host cell, some of them might be more mild, and they don't directly kill the host cell by lysis. Okay, so that's different types of bacteriophages. The first one is lytic, like T4 phage, or we call it virulent phage. These phages can only multiply within bacteria and kill the cells by lysis, including T4. Another type of phage called lysogenic or temperate phage. Temperate phage can either multiply via the lytic cycle, or sometimes they can enter a quiescent state. So they enter a quiescent state in the bacterial cell. They integrate their DNA into the bacterial DNA and replicate with the bacterial DNA. And for some reason, there might be some factors happened, and that can induce the phage to enter the lytic cycle again. That we call the induction. So different types of bacteriophage. I'll show you another video on lytic and lysogenic cycles of bacteriophages, and it will show you the detailed procedure of different life cycles.
Hey guys, how you doing? This is a video to show you the difference between the lytic and lysogenic cycles of how bacteriophages can reproduce. Just remember that this process, or both these processes, have only to do with bacteriophages, which is viruses that attack bacteria. It doesn't happen in animal viruses like the flu and other things. So basically, to kind of give you a sense of what's going on here, we see that here we have a bacteria. So this is the bacteria right here, and this is the chromosome of the bacteria, and this would be a bacteriophage. This is the virus that attacks the bacteria. So if we kind of see what's going to go on here, this particular virus is going to, as you can see, inject its DNA into the actual bacteria. And so now this bacteria has its own DNA, and it has the DNA from the actual bacteria. And so if we see what happens from this point on, we'll now see that the host DNA has been destroyed. The base of the phage DNA, the genes in the phage DNA, directed the host DNA to fall apart. And so now the only DNA that's left in the actual bacteria is the DNA from the virus, and I'm sure you can figure out what's going to happen from this point on. Now, of course, the ribosomes of the bacteria are going to be producing proteins. These proteins are viral proteins, and as you can tell here, the phage DNA is going to now code for the production of more viruses, and of course now these viruses are going to at some point explode from the cell, and that is basically the lytic cycle. And of course lytic means to lyse, or it means to cut or break open. So this results in the destruction of the bacteria, and that is basically the lytic cycle. So now we're going to go through the lysogenic cycle. So now this is the lysogenic cycle, and this differs in a couple ways, and I'll talk about them after. So the first part of this is the same, that basically a virus is going to attack the bacteria. And so what they didn't show here is that the virus would have landed here, it would have incorporated its DNA exactly the same as it did in the lytic cycle. But here's now the difference. In the lysogenic cycle, we see that instead of the DNA from the virus destroying the host DNA, it actually integrates itself into the chromosome of the host DNA. So now the difference is that, there is no destruction yet. So the beautiful thing for the virus is that, well, basically, it's not going to be making a lot of its babies, for lack of a better word, or offspring immediately, but this cell is going to start to divide, and it's going to divide zillions of times. And every time it divides, you can see that little teeny piece of DNA from the phage is going to, it's basically referred to as prophage DNA, is going to be carried from generation to generation. So now you have potentially millions, if not hundreds of millions of bacteria that all have this little piece of viral DNA. So at some point, when this host cell becomes either stressed or exposed to ultraviolet light or some kind of chemicals, a system called phage induction happens. Basically what happens, you'll see here that little piece of viral DNA pops out, and now basically the cell goes through the lytic cycle. So the second half of the lysogenic cycle is basically the lytic cycle. So what's going to happen? It's going to destroy the host DNA, it's going to start making the proteins, and there we go. So the difference between these systems is that in the lysogenic cycle, the chromosome or the nuclear DNA material from the virus incorporates into the actual phage DNA. And is carried generation to generation. So the bacteria doesn't make it, or the virus does not make as many offspring as quickly, but it is incorporated its DNA into many, many more cells, and thus giving the ability to potentially make much more offspring over a longer period of time. And so that is basically the difference between the lytic and lysogenic, and I hope you enjoyed it.
Okay, so the video shows us the different life cycles of lytic and lysogenic phages. We'll talk about these different life cycles in detail. First, the lytic phage. First, if the phage needs to infect the host cell, they need to recognize the host cell. On the cell wall, there are receptors, phage receptors. So the phage needs to fall down onto the receptors to recognize the cell for further infect the cell. And this step we call absorption. And the next step, the phage needs to inject its DNA or its genome into the host cell. So it penetrates through the membrane, through the cell wall. This we call penetration. You can see the phage genome now is inside. The next step is called biological synthesis. The phage DNA in the host cell will replicate to form a lot of copies of the DNA, the genome, and also translate into different protein molecules. And those different components will assemble together to form new viral particles. Those new viral particles finally mature and lyse the host cell and release. So this is a maturation and release. Finally, the host cell is lysed. So this is a lytic phage. So pretty simple. Well, for the lytic phage, it's useful because it can lyse the bacteria, which means that if you plate the phage on the agar carrying a lawn of bacteria, you culture the bacteria on the surface of a plate. This is a petri dish, the solid medium we learned before. And then you plate different dilutions of the phage, and if the dilution is proper, you will have single lytic plaque. So the phage will lyse the bacteria in this area. So you can see with your naked eyes that this area is clear. This area is clear. And this clear area or clear circle we call a plaque or phage plaque. And if you count the number of phage plaques, that indicates the number of phages. Through this plaque assay, we can determine the number of lytic phages, because each lytic phage will form one phage plaque on the petri dish. So this is the assay to quantify lytic phage. Okay, another type of phage we call the lysogenic phage or temperate phage. As you saw in the video, that this lysogenic phage are able to enter a non-lytic prophage state. This prophage means that the phage DNA is integrated into the bacterial DNA. So it links or insert its DNA into the bacterial genome, bacterial DNA, and become quiescent. And at this prophage state, the phage DNA will replicate together with the bacterial DNA. Okay, so this is the lysogenic phage. The bacteria carrying the prophages are termed lysogenic because a physiologic signal can trigger a lytic cycle, resulting in death of the host cell and liberation of many copies of the phage. So which means that the lysogenic phage can somehow enter the lytic cycle if it's triggered or induced, and this procedure we call induction. Okay, so this is lysogenic or temperate phage. Here's the procedure, lysogenic life cycle. Okay, first absorption and then penetration. The genome will be injected into the host cell. And there, the viral genome will form double-stranded and then insert into the bacterial DNA. And this we call prophage. And for some signal, the prophage will be kicked out, popped out, and duplicate, make a lot of copies, and also make components for the phage particle, and then release. And finally, it enters the lytic cycle and kills the host cell. This is the lysogenic life cycle. Okay. When some phage insert their DNA or the prophage into the bacterial chromosome, it somehow can give the bacteria some new properties. When the phage is inserted into the bacterial DNA, this bacteria might have some new properties, and this phenomenon we call lysogenic conversion. It means that the phage convert the bacteria and somehow contribute some properties to the bacteria. For example, it can spread of virulence factors, carry some virulence gene to the bacteria. And for example, it can carry some exotoxin genes or exoenzymes from one bacteria to another. So this we call lysogenic conversion. Okay, before we end our lecture today, I'd like to show you a story on bacteriophages. This is a story that people utilized bacteriophages for to fight against bacterial infection. Actually, before the time of antibiotics, that's early last century. So early last century, in 1930s or 1940s, some countries, some labs did pretty good job to study bacteriophages, because they know that some phage are lytic, and they think that it's reasonable to use a bacteriophage to infect the bacteria and use the phage to lyse the bacteria, which is a pathogen. So they can treat bacterial infection with phages. But then humans discovered antibiotics, and we realized that we can use antibiotics to treat bacterial infection. So that idea was given up for a long time. And now with the application of antibiotics, we are having more and more antibiotic resistant bacteria, and we have to increase the amount of antibiotics for treatment. And even though we increase the amount of antibiotics, some bacteria are still resistant to a lot of antibiotics. We cannot cure bacterial infection anymore. So that triggers the idea to come out again that we can use phages to fight against bacterial infection. So this is not a new idea, but it's coming back again. Now there are several big labs working on phage, and they want to find ways to cure some bacterial infection. So here is an example on application of phages to fight against bacterial infection. This is several years ago. At that time, this method was not approved by FDA, and it's not a regular procedure. So it's called an alternative procedure, alternative treatment. And antibiotic treatment is a standard treatment. And it's a story about two professors in UCSD, that's the University of California, San Diego. And those professors they are a couple. They went to Egypt for a tour, but unfortunately, the guy, Tom, got infected. And all the antibiotics they had at that time didn't work. So finally they tried phage, and it worked, and Tom was cured. So let's watch this video on their story. So phage against bacterial infection.
They were having a great time. They'd seen the pyramids, and they were on a boat headed up to Luxor. And as the boat was pulling into Luxor, Stephanie texted me and said that Tom had developed abdominal pain and was unable to eat, and she was worried that he developed food poisoning. She then texted me back and said, the pain is going through to his back. Could this be pancreatitis? What should I do? And I said, you need to get him to a hospital. The last thing you need is pancreatitis in the middle of Egypt. It's an organism we have a lot of respect for now. So after a little over a week in Germany, Stephanie was able to work with people here to get him flown to UCSD. And he was hospitalized here at UC San Diego just before Christmas.
I wasn't ready to let you go. And I came to you and I held your hand and I said, honey, they're doing everything that they can do. There's nothing that can kill this bug, so if you want to fight, you need to fight. But do you want me to kind of pursue some alternative therapies, and we'll leave no stone unturned. Around that time, a colleague of ours who's at UCSF said that a friend of hers had been treated with phage therapy and flew to Tbilisi, Georgia, where she had this miraculous cure. And I thought, wow, that sounds kind of far out, but wait. I remembered learning about phages when I was an undergrad at the University of Toronto. So I thought, wow, and I looked it up and I saw that it wasn't licensed and it certainly wasn't FDA approved, yet there were some papers saying that this was a promising area for the future. So I wrote Chip Schooley an email and I said, what do you think about phage therapy? And he said, what an interesting and intriguing idea. If you can help identify some phages that are going to be reactive against his isolate, I'm going to give it a whirl. We set on a phage hunt. Took just a couple weeks, but an international effort of researchers and people at the Navy Medical Research Unit came together to make this happen. We couldn't have done it without the UCSD team.
What they literally mean are bacteria, phage. They eat bacteria. And these are small viruses that are ubiquitous. We're loaded with bacteriophage, and on any given day there's a dance going on in our GI tracts of bacteriophage attacking bacteria. In the early part of the 20th century, there were a lot of efforts in France and what became the former Soviet Union to use these organisms as therapeutic agents in people with difficult to treat infections. Toward the late 30s and early 40s, there was less interest in this in the West because antibiotics began to emerge, and people in the West thought antibiotics would be able to take care of our problems with bacterial infection. There had not been a lot of experience with treating people with bacteriophage, and almost none giving it intravenously in the U.S. So we relied on some of the literature from Soviet Union, but a lot of it was really being worked out kind of on a first patient basis as we went, using a combination of the previous literature, our own intuition about how these phage would circulate, and advice from people who'd been thinking about it for a long time. Within about 48 hours, he woke up and turned to his daughter and said, "I love you."
Had there been phages available in the course of my illness when I first got ill, I wouldn't have been nearly as sick as it turned out I was.
There's a lot of interest in bacteriophage therapy. It's been gradually emerging over the last year or two as efforts to develop more traditional antibiotics have not really made the progress people had hoped. Bacteriophage have been kind of brought back off the shelf as a potential new approach to therapy. They're not simple to use. You have to develop a cocktail for each patient's own isolate. They seem to be relatively safe to give, but they're going to be difficult to develop from both the research perspective and also from the regulatory perspective. Now, the good news is we have awfully good tools now to do this, robotics and much more sophisticated molecular tools that enable this to be done. 10 or 15 years ago, it would have been impossible to contemplate doing this. And so I think, although there's a lot of research to be done, I think there are going to be a lot of clinical applications where this approach to therapy may be very beneficial to patients.
You can't emphasize enough how much I believe that phages really can change how we treat people in an economical way. So I see the third world and other places benefiting as much as we do. I mean, in my case, I can't tell you how much it costs to take care of me, much more than, I would like to believe. But if this can be done cheaply and really low-tech way, we can save millions of lives. And so it's, I think, the future of medicine.
This is the famous story using phage to cure bacterial infection, and Tom was actually the first patient successfully recovered through phage treatment. And nowadays, this story happened in UCSD, San Diego. And nowadays, there is one clinical trial in this institute which is approved by FDA. They are using other phages to fight against infection of Staphylococcus, and hopefully they will find better ways and to cure infection by antibiotic resistant strains. Okay, so this is bacteriophage. I guess we've finished the topic of today. We talked about the genetics, and we talked about their genetic components, and also special content in this area. Next time we'll talk about variation. So we'll talk about how we can change the genetic material of bacteria and how we can transform those microorganisms. Okay, that's all for today. Thank you.